I'm Lindo.
And I am Kurt Angelo.
So, yon, mitosis is a type of cell division in which one cell, which the mother, divides to produce two new cells, the daughters, that are genetically identical to itself.
Mitosis is to produce two identical daughter cells and the cell have the complete set of chromosomes. Because if the cell is too few or too many chromosomes, it will not function well.
The cell starts to break down some structures and build others up, setting the stage for division of the chromosomes. The chromosomes start to condense and making them easier to pull apart later on. The mitotic spindle begins to form. The spindle is a structure made of microtubules, strong fibers that are part of the cell. Its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. The nucleolus, a part of the nucleus where ribosomes are made and disappears. The chromosomes finishing condensing, so they are very compact. The nuclear envelope breaks down, releasing the chromosomes. The mitotic spindle grows more and some of the microtubules start to capture chromosomes. Microtubules can bind the chromosomes at the kinetochore, a patch of protein found on the centromere of each sister chromatid. Microtubules that bind a chromosomes are called kinetochore microtubules. Microtubules that don't bind to kinetochores can grab on the microtubules from the opposite pole, stabilizing the spindle. More microtubules extend from each centrosome towards the edge of the cell, forming a structure called the aster.
Good morning, everyone. We are here to discuss to you about metaphase. Metaphase is a process that separates the duplicated genetic material from parent cells. Process that duplicate the genetic material carried in the nucleus of the parent cell into two identical daughters. Microtubules pull the sister chromatid back and forth until they align along the equatorial plane or the equator of the cell. There is an important checkpoint in the middle of mitosis called the metaphase checkpoint, during which cell is ready to divide. All of this stage, the metaphase is the shortest stage of mitosis. It takes about 2 to 10 minutes. Prior to metaphase, protein formation called kinetochores, which are formed in centromere. Long protein filaments called kinetochores microtubules extended from poles on either end of the cell and attached to the kinetochore. And the chromosomes are pulled along by spindle apparatus. Once the cell has established that all of the chromosomes are properly aligned and attached, the kinetochores also are correctly attached, the cell enters the fourth phase of the mitosis, which is anaphase. And that's all.
Hello, hi mabuhay!
I'm Angel together with my partner,
Gwen, and we are going to talk about anaphase.
and we are one of the rapporteurs for today.
We can see in this illustration, the centromeres divides, and the sister chromatids of each chromosomes are pulled apart or disjoint. So in this illustration, we can see that it has spindle fibers that is connected to the chromosomes. These spindle fibers pulled apart the chromosomes, or simply as called the daughter chromosome. Anaphase. Anaphase is the stage of mitosis after the metaphase. So think that A in anaphase means away. Because the chromosomes are pulled away from each other. Anaphase is the shortest phase in mitosis. It takes only 2 to 3 minutes of the whole cell cycle. So for you to discuss it more further, my partner Angel Fatima will discuss the process that occurs in the anaphase.
This process is the spindle fiber will pull the chromatids to the pole. So this is the spindle fiber. Nag-aano siya ng force para ma-pull yung mga chromosomes. So what's the differences of chromosomes and chromatids? Chromosomes is part of the cell that contains the genes. Then, while chromatids is one of the usually paired and parallel strand of the duplicate chromosome. So I will draw the illustration of the two. As we can see, this is the illustration of the chromosomes, which it contains the genes. Then, while in chromatid, it is a single parallel pair. Then, sa unang proseso, ang ginagawa ng mga spindle fiber, itong mga guhit-guhit na dilaw, parang mga sinulid, pinupul nila yung mga violet, which is chromosomes na nagiging chromatids. Tapos, sa second process, the identical sisters slowly moving towards opposite pole. Yung identical sister na chromosome kanina, unti-unti silang pumupunta sa opposite pole at unti-unti nag-iiwalay. And the third process, identical means exactly the same or alike. Then the third process, anaphase is divided into 2 equal parts. So the final, naghiwalay na talaga yung sister chromosomes, being single chromosomes. That's all.
And today I'm going to discuss the fourth phase of mitosis, the telophase. Telophase is technically the final stage of mitosis. Its name derives from the Latin word telos, which means end. So, according to what I've researched, yung telophase daw ay yung opposite process na ginagawa ng prophase, wherein in this phase, the daughter chromosomes are incomplete opposite ends. Yung tinutukoy ko po sa ito at ito. So, nabanggit ko po kanina, sister chromatids. Sister chromatids po, yun po yung chromosome that results from the separation of sister chromatids during cell division. It contains the complete genetic information ng parent cell na tinaglay nila. Spindle fibers ng nila sa kanila papunta sa post na ito, which is ito po yun, ay mawawala. Sister chromatids po ay magsisimulang ma-decondense or ma-decoy para maging long and thin strands ng DNA na tinatawag na chromatin. Ang chromatin po yung substance distributed in the nucleus of a cell that condense to form chromosomes during cell division. It consists mainly of DNA and proteins called histones. After that, new nuclei are forming on each side to make new cells. Dito, yung nuclei ay mare-reform along with nucleoli na magkakos ng reformation ng nuclear membrane na papalibot sa chromosomes on both sides. There will be 2 identical cells na mayroong nuclei each, and sa human body, yung cells na yan ay parehong nagko-compose ng 46 chromosomes.